grade 12. Welcome back to Maths and Science series Life Sciences with Spongile. So, on the first part is the introduction to the DNA code of life topic. I gave you some basic terminology that you will hear me talk about and say throughout the topic parts that I will be doing. So now we are going to be doing the structure of a cell. The structure of a cell is something that we have been doing for the previous grades. We were labeling specifically the plant cell and the animal cell. And now we're looking deeper into the part that we labeled as the nucleus. So we have the nucleus being that hollow ball that you see there that is so big and purple. I made it cute for you to see clearly. And then I have the ribosomes on my cell there. And those are the red dots that you see on your endoplasmic reticulum. So the endoplasmic reticulum, it is that blue sap inside there. That blue sap, we call it the endoplasmic reticulum. And then we have the chromatin networks, those looking like spaghettis right in there in our nucleus, and they are surrounded by the nucleoplasm, which is the jelly-like fluid. I told you it is found in the nucleoid. And then we have the nuclear envelope. The nuclear envelope, it is the membrane that is surrounding our endoplasmic reticulum. Okay, and then we have the nucleopore, which are the scientific gaps between our nuclear envelopes. I hope we're clear on that. And further down, I drew that arrow to show you that we are concentrating on the nucleus. So that is the nucleus, and those are the chromatin networks. And then they become those funny X-like shapes when they condense. So they condense and then they become what we call chromosomes. So for you to get chromosomes, the chromatin network will have to condense. After it has condensed, it will form the chromosomes. And then we know that our chromosome is made up of a chromatid and another chromatid. They are both joined by the centromere for them to be called altogether a chromosome. And then what we are telling you now is that if you take a microscope and you take a condensed chromatin network, which is a chromosome, and you look into one chromatid, this is the shape you will see. And this molecule is called the DNA molecule. That's how you see the DNA molecule. It looks like two strands that are twisting to form the shape of the DNA molecule, which we'll get further into when we talk about the structure of DNA, which is called double helix. Love you guys a lot. Don't forget to like, comment, and subscribe if you haven't, because it means a lot to us and it motivates us to keep doing more. And please share with your peers so they may also find this easy peasy knowledge that you're getting that will be useful for your final examinations. Thank you very much for being with us. See you next time.